we will have today's Theo take us through the first topic, which is the quality check and data cleanup. All right, so we do have a lot of content. So Teddy, please take us away. Thank you.
Right, first thing first, can you see my screen?
I can see your screen.
All right, fantastic.
Yeah, you are a little soft though. Is that possible?
I'm a little soft? All right, I think it's just the mic. I just need to bring myself closer.
Yes.
Yeah, okay, is this better now? Should I increase the volume?
This is better.
Much better? Okay, I hope I can maintain this level of volume. All right, so let us begin. Hi viewers, thank you for taking your time to attend this masterclass. As you know, I'm Teddy, a Fluidigm FCS with 3 years of CyTOF 2 and Helios user experience. So for today, I'll be a presenter and moderator, and I do apologize in advance as you see what happened just now, that we might not be able to answer all your questions that everyone has. And if I do miss any, please do not be shy. Please just put it on there, and I'll promise that I'll get back to everyone via an email message, right? So now back to the section where I'll be presenting, and I'll take about 10 to 15 minutes. But so let us now begin to look at the new method of data cleanup that's used with our PubSetter software. This will help the CyTOF users to clean up their data. First, they will do a quality check, right? Then we'll look at how we clean up the data before moving on to analysis, where Andrea will lead us into a fantastic journey of what FCS 7 can do for you. Now, in this section, we'll cover 2 points. Basically, we'll review on how to assess the mass cytometry data for sample quality. Then we will look at how to use the Gerson parameters to identify live single cell before moving on. This Gerson parameters method differs from the pre-2021 method that used to identify live and single cells. And you'll see the difference in there, and I'll elaborate a little bit more. So now let us begin with the data quality assessment. This should be always the first step before we commence any cleanup and population gating. From there, we can scrutinize if the acquired sample is good for in-depth analysis. At this point, there is a couple of steps to go through. You can see that we will begin with the raw FAS file. We will check 151 EU and then 153 EU median signals intensity for EQ beats. We check the events rate, and we will start to normalize the files. Once everything is okay, normalization begins. When normalization begins, from there, we will assess the 140 CE and versus the 191 DNA iridium plot. And then from there, we will finally identify and examine the signal versus time plots before we move on to our data cleanup. So this is when you're doing this with an ungated raw file, right? So you have to look at 151 and 113 EU to make sure that the medium signal intensity should be more than 1000 for the beats prior to normalization. This is usually done when you are looking at QC for your EQ beats QC, or you can actually use it to look at the EQ beats that's inside your sample as well. But I recommend it personally with your EQ beats QC. If it's lower than 1000, please look at your system. You might need to do a bit of a wash on the front end to make sure everything is up in order. It might be a bit dirty, so you just clean it up and it should be ready for your sample run. Now let's check the CVs, right, for 151 and 153. As you can see, for those who are sharp eye, at this section here where my mouse is pointing, those that doesn't have very high CVs that doesn't really pass is when you look at this particular section here. It's actually for your EQ beats, it's more laid out and not really condensed, whereas this is the good one that you're looking at. This is the one that's okay. It's condensed here, and you have very, very low CVs in this. This may be why do you have this in the end? It's not okay. It's most probably due to the cones that's being dirty, resulting in a broader CV. So if you do this, do try to clean your clones before you rerun the whole thing as well. Both when you clean the clones, remember it's both skimmer and reducer, and this can help you to tighten the CV thereafter. Now, when you check the event rate, right, time versus 101 iridium, you have to check on the raw FCS file, and you should be looking at ungated events. While the event rate should be closely monitored during sample acquisition, we can also use this plot to check for a closer inspection. Remember that the high events rates increase the number of ion fusions. This is important, right? Don't run too fast. Therefore, as users, we should always aim, right? As an experienced user, I'm letting you also know that we should usually aim for 300 events per second or lower, because this acquisition rate produces less ion cloud fusion, making the data more usable in the downstream analysis, and also not forgetting that the optimal rate, optimal event rate will depend on the sample type and also should be determined empirically. That means whether how much you want to run, how much you want to gather, you should look at what type of samples you are running. Now, this step is accessible after the files has been normalized. Once you're normalized, you do a quick view of 140 cerium and 1191 iridium, and it should be ideally inclusive of low cell bits doublets. There should be no or low iridium signal, and it should be around the power of 3. You can see that it's around this section here when you're doing 191. And when you are checking signal over time, do check the 140,191. Make sure the other abundance marker versus time, they should be constant. There shouldn't have any negative. The negative should be low or not at all. Ideally, there should be separation between the positive and negative signal like this. You can see that it's really clean cut between both, right? Positive and negative signal. So this is a overall good data view. A consistent this indicates, when you look at this data, with my experience, and a lot of users will tell you that there is actually no interruptions at all. With interruptions, what do you mean? That means there is no sample introduction issues such as clogging, clumping, sputtering, or linking, right? If the negative signals are close to 10, it means that the sample may have a large amount of debris. This negative center should be close to 10. If the sample has a high amount of debris, dead cells, non-specific staining, the residual unbound antibody, the negative signal can actually be much higher. So please take note about that. How do you prevent that? You just do your proper sample cleanup and make sure they go through the processes. Right. Another thing to observe for high quality data features are the EQ beats, live date, the DNA intercalator signal. Signal from metals such as DNA, live coding, barcoding, antibodies should be within the range of the instruments. If the signal appears out or to max out and lies along the upper y-axis, titrate the reagent. Make sure that the even the negative markers should have some signal in the channel, but it's usually less than 10. If the signal is 0, there might be an issue with the reagents. So just keep note about it, right? That's within this particular isolated, you should get them within this particular section. That should not be off the dynamic range. Now, lastly, the beats doublet should be close to about 1%, a few percent at most. A higher amount of beats cell aggregates could indicate there's too high concentration, high debris, or clumping. The amount of debris and dead cell will vary dependently on the sample type and disease status. High quality data from healthy PUNGs should be around 85% live. Single cell suspensions derived from tissue or disease or treated samples such as cancerous will end up having higher debris or dead cells. Researchers may need to investigate proper sample preparation techniques. That means you improve on your techniques and protocols to achieve higher quality data. Just take note about that. So moving on, now we'll drop in some time in regards to the area where we look at the strategy for the data cleanup. Okay, the first step of the data cleanup is to remove unwanted events. Then after that, why we use this is to remove unwanted events, and also the data cleanup is to use to remove dead cells in context, right? Dead cells, debris, and then normalization bit doublet, so that and the ion cloud fusion, so that you can start to jump into the analysis. They must be done manually prior to downstream data analysis before other algorithms and software. Okay. So this particular method of cleanup is called the GDP cleanup, which is the Gaussian discrimination parameters. It's been developed by Fluidigm and Verity Health Software, which was meant to be used with the MDIPA path setter or MDIPA assay, which is short for MaxPath Direct Immune Profiling Assays. Okay. This particular strategy uses you can see here 140 CE 191 event length and viability, Gaussian discriminant parameters such as the center, width, offset, and residue. It does a proper cleanup than the older, it do a better cleanup than the older ones, which just gates on the events of the DNA and then just your live dead cells. So it goes more in depth, and Andrea can show it in more details later on, but I'll just walk you through in regards to this at this point in time. So what are Gaussian parameters? They are determined by the Halos event detection. You can read through this. It's a bit crowded on this slide, but I just want to let you know that they have single self-generated ion cloud transitions that have similar values for GDP, while debris, aggregates, and ion cloud fusions fall outside this range, and we utilize this range within the center offset residue and center offset width and residue to start to isolate and look at the files that the cells that you want to investigate, so that we can take the information out from there. Now, how does the Gaussian fit is calculated? It's based on this. We can go into some other details later, but at this moment, you just understand that this based on this calculation, and that's how we determine it and to fit within the Gaussian curve and to take the information out. If you want, we can read more about this method through background. Okay, at all at 2020. Now we will start, the procedure is this, straightforward. We'll go for 1 to 9. I'll go step by step as you can see. So the main aim from 2 to 6, you want to understand that is to remove debris aggregated cloud fusion. Then we will start to look at removal of the date, live date to identify the live date, the DNA and 1 and DNA 2. Why do we use DNA 1, DNA 2? It's basically to identify cells in mass cytometry because we do not have light, so we don't have forward scatter or side scatter to determine whether is it a cell. We use DNA, which can be tagged into the major groove of the DNA of each individual cells, so that when we run through the mass cytometer, we know that when we do this 1I1, 1I3, we can use it to identify a cell. Right, so this is a summary of how it looks like, the outline. We will start with all these parameters that's going there. So after this, the DNA will move into downstream analysis. We'll start getting to ID populations, and we can move into higher dimension analysis, which Andrea will pick it up and run with it later. Now, in regards to this, we first look at in details, right? We look at CE versus time. We remove the beats. We need to remove the beats because it doesn't link up anything that we need to know. So we just adjust in this image here. We select what is within this particular section. We just ignore what's up here so that we can look at the beats. Then we move into residue over time. We want what's right in the center. We adjust the cleanup residue to determine the denser of events with the mid-range intensity. That's usually around this range of 10 to the power of 2. This is in arcsine, yeah. This is in linear. So you can actually, when I say arcsine, it means the scales. So that when you do this, it actually cleans up this particular area right here. You select. From there, you go further to look at the center. This is a bit off. It has to be a bit more centralized in this area. But what I want you to understand that we select the center, we remove what's on top and what is below, right? Because we can see that they are basically just either doublets, debris, or anything that's there. And then we move in offset versus time. We do the same thing as well to look at the parameters in offset. When we move on to width, and we clean out the width so that we can get what's right there in the center. And then finally, we move into event time versus length, and this helps to event length versus time. So we help to eliminate ion cloud fusions from this. Okay, so you might need to adjust scale and resolution for better visualization. Please take note about this. And moving on, we will finally move into two date cells here. We have two options. One of the one that is commonly used with the MaxPar direct immune profiling assay is rhodium, which is 103 RH. And this permeables the cells, indicates double-stranded DNA and fixed into the fixed perm, right? So if you're using this, you can use this particular method, our rhodium that's here, and you do the cleanup and you select what is in there, okay? The best practice is to use method calculating. So this helps you can count it using your Terracan Blue, using the manual cytometer to also help you to identify and verify that these are the live cells that you are looking at. So the second one is another option people use is cisplatin, which is range of 195 to 198, depending on which one that you use. So this helps you to also identify which is here. The only thing that when you need to be concerned when you're using cisplatin and then you are looking at mostly granulocytes such as monocytes and macrophages and dendritic cells, you may want to skip this cisplatin option, but look at this way of gating instead. Why? Because it turns out that macrophages and dendritic cells, due to their larger shape, right, of the mononuclear cells, they tend to pick up a little bit more cisplatin than anything else inside the rest of the cells. So you just need to be aware of this. You might want to be a little bit liberal in your gating if you're looking at macrophages in this section. After that, we move on to DNA, where we clean up and then look at the denser intensity right there. Okay, for your DNA 191. 191, remember that naturally occurring in nature, maybe about 38%. And then after that, we reflected with, 32%, where in DNA 2, 193, it will be about 68%. That's why we use both. And then from there, you will end up capturing 98 to 99% of the cells. So, and this is in summary, a very quick way of how to do it. So it's a bit more steps, but it's definitely cleaner. I've been using it to help customers, and we see much more cleaner areas where we tend to gate. Okay, so this is all that there is. I don't want to drag and pull out any more time from Andrea here. Please let me know if you have any questions in the question and answer section. Okay, we are You can go straight into questions and answers right now. Anyone has any questions?
Thank you for trying to help me make for the time.
No problem.
Yeah, looks like I think we can proceed on to Andreas, and then we can take in any questions on that analysis together.
Okay. Can I just answer to Sujong?
Yes,
Questions?
I see that we have a question over here on data.
Yeah. She asked how many cells is left after cleaning. It really depends how much you get to start with. Please do, if you want to know, it really depends on what you're also looking at and also your sample. It will say that in the end, if your sample is bad to start with, you're going to get a bad resolution back in the end, maybe just only a couple of 100,000s, right? But if it's good, it can be more. So if you have bad samples, you may want to capture more data so that you can have a look, and the cleanup will be much better. We can talk more about this if you have concerns about what kind of samples you're using. We can bring it offline and we can discuss more. Just drop me an email and I will come back to you on this because every sample is different. Now I will pass the floor to Andrea.
So, Andrea, can you turn?
Yeah. Can you see my screen?
Yes, we can see your screen. So just do a quick introduction. So we have Andrea Vel from Novo. He's taking on the next topic, which is high-dimensional data analysis for mass cytometry. This session is lengthy. It will take about an hour, right? And we would invite everybody to, again, put in your questions through the Q&A tab. So it's a lot easier for us to identify the questions and share it with the audience as well. All right. If you were to put it in the chat, I'm unable to share it with the audience. All right. So FCS, let me just do a quick introduction about FCS Express as well. FCS Express is one of the leading tools on the market right now for getting results from cytometry data sets. So with our close partnership with Novo software, we are able to further accelerate our researchers and our users moving from the raw data files to final usable data and data results in an easy to use and efficient manner. So welcome, Andrea, and we'll leave our audience in your good hands.
Great, thanks. Thanks for this nice introduction, and thanks for giving me this opportunity. So my name is Andrea Valle, and I'm technical application specialist at Novo software. And let's focus on the screen. Now I will switch off my camera for a while. I'm technical application specialist at Novo software, and yeah, I'm going to present you FCS Express, and we will focus on high dimensional data analysis. Maybe some or most of you are new to FCS Express, so let me give you a flavor of what we want to do with FCS Express before moving on with the practical session. So the Novo software, which is the company which is developing FCS Express, is based in California, in the sunny Pasadena. We are about 25 people specifically devoted to develop FCS Express, so we're not doing anything else. We have 10 application specialists as me. We're spread in 5 different time zones around the world. So this is great to support you, because whenever you have any question, just write us, and we will usually being able to reply in a couple of hours. Then we have 10 people developing the software, so mathematician, statistician, developers. And then we have 5 people in the office dealing with licenses, renewal, and sales, and all those stuff. Now we have a wide variety of customers, of users who spread along, like CRO, big pharma, private company, academic, hospital, clinicians. So we also have an IVD version, so for people dealing with diagnostics. So as you can imagine, we can really fulfill the needs of a lot of different customers. Now, the history of the Novo software started in 1988. So it's now 23 years that we are dealing with data analysis. Okay, so we are really keen on this. And again, we're not doing anything else. Now, the latest release, it's named version 7, so FCS Express 7, has been released in August 2019. We're not going to change the name, okay? From the marketing point of view, it's version 7 and it will be version 7 for a while, but we are really, really, really keen in releasing updates. So every 2-3 months, we are releasing new features in FCS Express. And this is, of course, thanks to all our users that provide us feedback. Then on this slide, just let me mention that, again, we have an edition for people dealing with academic or anyway research, okay, not clinical, I will say, and an edition for people dealing with clinical. So it's good to mention. Now, let me clean. Yes, here we go. So we are super excited to be here today and to work with Fluidigm. And when we started this collaboration, we really realized that Fluidigm and the Novo software are going to the same direction. So Fluidigm is providing you with cutting edge instruments. Okay, as you saw in this presentation, and with the Novo software with FCS Express, we want to do the same for the data analysis part. So we are really going to the same direction. And thanks to this, we are super happy to provide a free 1-year license for FCS Express with any instrument. Okay, so if you have an instrument, if you purchase an instrument, in this case, you see I've used the old, let's say, picture of the instrument, but of course it will be true also for the XT, no worry. And you will receive a copy of FCS Express. So today it's also a good opportunity to see why FCS Express is so special. And this is probably the most important slide that I'm going to show you today. So at the left, you can see the standard workflow in data analysis. At the right, you can see the FCS Express way of doing data analysis. Now, at the left, you have your data, you acquire your data on the instrument, and then you start creating plots and gate. Okay, so this is what you can do with every software on the market, even with the software which is like 10 years old. Plots and gates, that's it. Okay, you can retrieve numbers, of course, but it's really hard, if not impossible, okay, to achieve the final results with the usual cytometry software, data analysis software. Why this? Because you need to rely on external software. You need to do a bunch of unneeded steps between the data and the final results. For example, you need to copy and paste in PowerPoint or in Photoshop. You need to export or copy and paste in Excel or GraphPad to create some charts. You need to run high dimensional data analysis. This is the cool topic of today. You need to run this type of algorithm with plugins or maybe with external software like R. And this is time consuming. It's annoying because every time you need to rely on external software. And even worse, if you want to, let's say that you spend time to create your final results, you're super happy. And then maybe you realize that one of your gates of your analysis should be a little bit different. Okay, so you need to go back, move the gate, and then re-copy and paste, re-export, create the charts, copy and paste, rerun. It's a never-ending story. It's time-consuming. It's annoying. It's not efficient. So it's what we want to avoid with FCS Express. This is why we are here today. So on the right, you see FCS Express. What we want to do, what we're working on since 23 years is to be able to integrate both data analysis, so plots and gate and all the standard tools and cytometry with data reporting and calculation. So you can create heat map, you can create charts, you can create high throughput screening plates, you can make calculation, you can create formula in Excel, okay, with your site of data. You can achieve the final result, the final goal inside the layout. Now you see here, I have an example, final results. So let me bring on the screen a layout from FCS Express. So what you see here, it's not PowerPoint, it's FCS Express. We will see a little bit of the user interface in a second, but this is FCS Express. It's similar to PowerPoint. You see we have a ribbon bar at the top, okay? The ribbon bar, it's similar to PowerPoint. For example, if I go to the text, okay, you see here, I can format the text that I have on my page. If I go to design, I can change the size, I can change the orientation of my page. So what you see here, it's one page among many in my layout. You can see here that I do have many pages, okay? This is 1, this is 2, this is 3, this is 4. I can scroll through pages exactly as I would do in PowerPoint, okay? I call pages, you can call them like slide. It's up to you. So what you see here, it's like a slide. Let me zoom out a little bit so that you can see it entirely. So usually in this case, you want to start with a cleanup page. So here you see I have all my cleanup plots. Okay, so we will see how to create this. I'm just showing you that you can combine in a page, as you can see here, plots. You can combine text boxes, you can bind the hierarchy, and then you can move on. For example, let me open the CD8 page. Here I have a CD8 page. I have my gate hierarchy here. I have some statistic here. You see some table, okay? And I have some plot, well organized as I want. If I want this plot to be here, that's fine. If I want to move also text box, that's fine. If I want to make it larger, that's fine. Okay, you have full control on the organization because again, this is data analysis and data reporting. And as I said, let me go to the last pages. Okay, last 2 pages, we have a spreadsheet. This is like Excel. Okay, you see we have different columns, different rows. Okay, and you have the ability to create formula. We have a lot of formula here. You see, we try to be consistent with Excel. You can create statistical formula. You can create mathematical formula. Okay, so if you want to calculate an ANOVA, t-test, this is FCS Express, data analysis and data reporting. And of course, we have charts. We have many charts available. Okay, you see here we have bar, pie, we have box and whisker, violin plot, beeswax plots. And in this case, I'm showing you a very simple bar plot. And I want to show you that in FCS Express, everything is live updating. So what you see on the screen now, it's a bar plot depicting a sort of summary. You see, I have a bunch of population of interest, okay, with all the statistics. And what I've done was to graph those statistics, those percentages in a bar. And as you can see here, I have a plot. This plot can be in another page, or it can be in this page. It doesn't matter. This plot, it's a gate on CD3. And as soon as I will adjust this gate, you will see that few bars will update in real time. Okay, those bars that update in real time are all the T cells population. So in FCS Express, you have live updating capability inside the software. This is true also. Let me go back to the CD8. Okay, if I move this, you can see that every other plots and also in every other page updates live. Okay, so this is the idea of FCS Express, having data analysis and data reporting and graphing tools all embedded into the same platform, a PowerPoint-like platform. So it's a very user-friendly interface. So let me close this and let me move on the first part of the presentation. So I want to show you how to create plots and gates and giving you the idea about how easy it is to use FCS Express. Okay, so let me open a new page, a new layout. So this is a blank, empty, clean layout of FCS Express. Okay, you have the ribbon bar at the top, as you can see here. And the ribbon bar, it's really a copy of what you have in PowerPoint. You see it's subdivided in different tabs. If I open the File tab, the first one on the left, you will see that it's really similar to PowerPoint. So you can open, you can close, you can export. If you want, you can export your FCS Express layout directly to PDF, to PowerPoint as a high-resolution picture. Okay, you don't need to do a single copy and paste. Copy and paste to PowerPoint, it's something that people are doing a lot if they do not have FCS Express, and it's really time consuming, it's annoying. In this case, you can do all what you want in FCS Express, and at the very end, with a simple click, you can export to PowerPoint directly. Then, this is my page. You already know the design tab. So if you want to change the orientation or the margin or the size, just go here. And for example, switch to landscape. Okay. We also have a zoom at the bottom left. You see here, I can zoom in and zoom out. So just to get you started, okay, I will insert a text box. Okay, let's say that click on text box, draw it as you will do in PowerPoint. So there is nothing to be scared. Okay, this is my first experiment. Okay, so this text box can be, oh, sorry. Here we go. This text box can be resized, can be moved around. We also have a text tab. Okay, you can change the font, the size, all the basic word editor options are there. Then you can also, of course, insert other objects. You see, you can insert shapes, you can insert the gate hierarchy, you can insert picture, maybe the logo of your company, the logo of your institution, of your university. We have an integrated spreadsheet, okay, that allows you to make calculation. It's the same spreadsheet that we saw in the example that we saw before. And you have access to all the plots that you want. For example, if you want to create a density, just click on density and draw it. And now the software is asking me, well, Andrea, which file do you want to load? And you see here, I have a bunch of files, okay? Those are total blood. I will click on, they are like medium size. You see 250 megabyte. Okay, I will click on open, and now the file is loaded. Now, this is not a picture, it's a plot, it's an interactive object. Okay, so I can resize it, I can move it around if I want. And of course, if I click on the axis, I can access all the parameters saved by the machine. And you see, it's a long list. Okay, it's really great because you can access a lot of markers here. Now, especially with this instrument, since you have a lot of parameters, it's good to know that you can type here. So for example, if I want, you see, I can type bead and I can filter and I can see all the parameters that are including that word. So for example, we want to start with a cleanup gate, as we saw before, by displaying beads, okay, 140, and time on the x-axis. Okay, and in this case, I'm super happy to show you how to create the first gate. Okay, so just go to the gating tab. You see we have a bunch of gates here. Just to show you, if you draw an ellipse, just click on ellipse. Okay, it's very simple, and then type the name. If you want a freeform gate, draw a freeform gate and type the name. You can also change the color. Okay, whenever you want to delete a gate, simply click delete on your keyboards and you will be able to delete it. Okay, now in this case, instead of ellipse or freeform gate, it's much better to create a rectangle. Okay, so we'll create a rectangle here. Okay, and let's say cleanup 1. Okay, red, click OK. And in this case, you can resize the rectangle from the left to the right. You can also resize it from the top to the bottom, and so on. There is also another gate type that I want to mention, which might be really useful for this type of cleanup gate. You see the rectangle, as you might imagine, can be resized in all directions. Okay, let me delete it. Let me show you also what you can do if you create a marker, which is a 1D gate. Okay, I can draw it by dragging and dropping my mouse from the top to the bottom. Okay, this will be cleanup 1. Okay, what's the advantage in this case is that I do not need to resize it from the left to the right because it automatically includes everything. I just need to adjust it from vertically, let's say. So it might be good. Of course, if it happened that you have some fluidic like interruption, okay, then it's much better to use the rectangle and gate out that interruption. But otherwise, you see the marker, it's pretty useful. Okay, so I have a layout here already open. Okay, this is exactly the same cleanup strategy that we saw before. And as you can see here, usually you start with time versus 140 CE bits, and then you have time and residual. Okay, and then you have time and center. So you go through all those cleanup gates one after the other. So we are here. Okay, how can we create plots number 2? Well, let me go back. So it's really easy. We want, of course, plot number 2 to be gated on the cleanup 1, right? So it's very easy. Use drag and drop. So what I'm going to do now, it's dragging and dropping this gate out and release it here. And we will create a new plot. Okay, so drag and drop. Here we go. We have a brand new plot, which is an exact replica than the first one, but it's gated. And you see that in FCS Express, everything is live updating. It's super, super, super fast. So plots 2, it's identical to plot 1, same size, same aesthetics, but it's gated. So now we should go here and simply change the parameter on the y-axis to be the residual and create another gate. Here we go. Clean up 2. Okay, and then drag and drop. And then in this case, we want the center. Okay, and again, clean up 3 in this case. So first of all, you see that it's really easy to use. Okay, so drag and drop in this case, it's an example on how to use drag and drop to create a new plot. But you can do a lot of stuff with drag and drop. We will see this also later on. But what I want to show you is that it's really easy to use. It's user-friendly. The PowerPoint-like interface allows you to feel yourself in a user-friendly and very familiar environment. You don't need to learn everything from scratch. Okay, so if you want to change the page setup, if you want to go to insert a new object, if you want to go, you see here in the home tab, I can select my plots and say align them to the top. Okay, arrange them like distributing object horizontally so that they are equally spaced. Okay, everything is laid down on my page as I want with live updating capability. You see, I can change the position of my gates and everything is updating live. This is not only true for 3 plots and 1 page. Remember, I was dealing with like a 15 pages layout with a lot of files. Now, what you're creating with FCS Express, it's, and this is really good to mention, it's a template. Okay, a template, it's something that you can use to analyze multiple data. So you see here, I have multiple files of interest from this experiment. I have drawn my, let's say, gating strategy, plots and title and whatever using file number 1, but I don't need to repeat this for every single file. Okay, I can go to file number 2 and see how it looks. I can go to file number 3, file number 4. Okay, I can do this. So I can use my analysis as a template. And of course, you can adjust gates sample by sample. You can make them, you can make gate consistent among all samples. It's up to you. You have all that flexibility. But the main point is that you don't need to create multiple pages, multiple plots, multiple gates if you're dealing with multiple files. This is really time saving. I'm not going to create the full cleaning up hierarchy you see here. Okay, so far we create plots number 1, 2, and 3, but it's just a matter of dragging and dropping, changing the y-axis and so on. So it's really easy to handle. What I want to do in this case is to show you final plots just to display some other markers. For example, let's say CD45 and CD3 here. Okay, you can create another gate like ellipse. Let's call this CD3. Okay, and then you can move on like CD3 and display CD4, CD8. Here we go. Okay, of course you can retrieve statistic. Okay, so let's say that you have a gate here on CD8. Okay, one way to retrieve statistic from a plot is right-click statistic, and you see now I have a nice table with all the statistic from this plot. And this table is of course live updating, so it doesn't matter which gate I'm changing, you see that the statistic updates live. I can also move the parental gate, still it's updating live. Okay, this is as I said, it's a medium-sized file. It's a quarter of a gigabyte, but even if you have like 1 GB files, it's okay. Okay, so let me close this. Let me go back to my presentation. So I want to switch to the high-dimensional data analysis part. So what we saw so far was a very small, short introduction on FCS Express, just to show you that it's really user-friendly and PowerPoint-like. And then I showed you how to get started. So how to insert the plots, how to create your first gate. And then with drag and drop, you can create the second plots, and so on, and so on, and so on. We also saw the cleanup gates all done, remember, this layout. And what I want to do is to switch now to the high dimensional. So first of all, why we need high dimensional? Well, because you have really a lot of parameters. Okay, parameters are dimensions. If you have like a 2-color experiment, you can plot on a 2D plot, so you have 2 dimensions. If you have 3 colors, you have 3 markers, so you will need a 3D plot. But then what happens if you have 4, 5, 6? Or what happens if you have like 52? Okay, so you cannot create a 52-dimension plot, not even a 4-dimension plot, of course. The only thing you can do is to open a multitude of 2D plots. But this is, first, time-consuming, as you can see here. You have a lot of plots, and it's not efficient because it doesn't matter if you have like all the plots open on your screen. How can you analyze this type of this amount of data? So even if you go through them one by one, it will never be, let's say, efficient in terms of data analysis, because you're still looking at your data at 2 dimensions at a time. Now, if you think about your FCS file generated by Helios as a matrix, okay, you can imagine this matrix to have cells in rows and dimensions, so markers, okay, in columns. Okay, you in this case, I'm just have like 20 parameters, but you will have like 10 or 30 or 40. Now, we need to simplify this table. We need to simplify the data. So you have 2 options: dimensionality reduction, which means reducing the number of columns, or clustering, which is reducing the number of rows. Very popular algorithms like Disney and UMAP and Optimize Disney and Principal Component belong to the first family, dimensionality reduction. Other very popular algorithms like k-means clustering, SPADE, Finograph flow sum belong to the clustering family. Okay, today we will see an example of dimensionality reduction, and if we have time, also clustering with flow sum. Now, when you're speaking about when you speak about dimensionality reduction, high dimensional data analysis in cytometry, usually you go through a workflow. It's not like loading your data and running Disney. That's it. It's not like that. Okay. Usually you have some pre-processing steps, which are really, really, really important. Okay, pre-processing include a lot of different steps, like quality control, actually. Okay, data cleaning, as we saw. Pre-processing might include like data merging. Okay, if you want to run a Disney or a UMAP or a flow sum on multiple files, you're not going to do that file by file. You're going to do that on multiple files combined together, merged, concatenated. Okay, pre-processing steps are selecting the population of interest. Okay, selecting the parameter of interest. Okay, you might say, I don't want to make the calculation on all the parameters. Okay, for example, you might say, I know that I have some empty channel. I don't want to include empty channel in my calculation, in my Disney calculation, because it will just bring noise. Okay, so selecting the parameter is really important. Then, super important, parameter scaling. Okay, you may know that in flow cytometry, for example, you usually use like B exponential scale, hyperlog scale. In mass cytometry, use arc-sine scale. Okay. In the old ages, we used like log scale, like in the 90s. Okay, for flow cytometry. Well, usually you don't use linear scale. Okay, there are few exceptions, few parameters and few assays that require linear scale, but usually you apply a different scale. But anyway, the question is, you need to be careful about which scale you want to use: linear, log, arc-sine, B exponential, whatever. You might want to normalize, okay, your parameter. You might want to downsample. So those are all the pre-processing steps. And then you have the algorithm itself. Okay, this is the fancy part: t-SNE, UMAP, FlowSum, whatever. And then you have the data reporting. So we realized at the Novo software that people, when people need to do this without FCS Express, they are not able to do this within the same software. They need to use the data analysis software to do something, then they need to go to R, then they need to go to GraphPad Prism, then they need to go to PowerPoint, to Photoshop, then they export, then they go back to their data analysis software. It's a never-ending story. What we decided to do in FCS Express was to giving you the ability to run this type of workflow from the beginning to the end entirely in FCS Express, thanks to a new features called pipeline. Okay, so you can combine, you can recreate that workflow, building that workflow step by step, like downsampling, scaling, normalization, clustering, dimension height reduction, one by one. This is pipeline. Pipeline are very well known in the bioinformatics, in the mathematician, among like programmers, developers. So they are very, very, very well diffused, but they were not diffused in the cytometry community. So this is why we are super happy to provide you with this tool to analyze your mass cytometry data. So the pipeline, it's a set of data processing elements. Usually they are like mathematical calculation. Okay, and the key point is that first, they are connected in series. So you have step 1, then you go to step 2, then you go to step 3, then you go to step 4. So they are executed one by one. Okay, the second key point is that usually, most of the time, the output of one step, okay, so the data generated by step 1A, in this case, it's usually the input of step B. And the output of step B is the input of C, and so on and so on and so on. Now, as I said, pipelines are very popular in the mathematical and statistical field. So if you think, popular algorithms like SPADE and FlowSum and t-SNE, they are pipeline themselves, but in standard software, okay, not in FCS Express, but in other software, they are run as a standalone algorithm. So in other software, you can run t-SNE, or you can run FlowSum, or you can run UMAP, and so on. If you want to run all, you might probably need to go through a bunch of intermediate steps, like running t-SNE, then export the data with t-SNE embedded, then re-import the data, then run FlowSum, then save the data with FlowSum embedded. It's a long way. In FCS Express, you can run everything inside the software, and you can combine them as you want. You can also create very, very big pipelines, so in which a small pipeline can be a part of a larger pipeline. So pipeline usually, why they are so powerful? Because they are flexible, because they can be combined, because they are reproducible. Okay, and you might say, well, why we didn't start using pipeline like 10 or 20 years ago? Well, because pipeline usually looks like this. Okay, looks like this. So they are really like programming language. And it took like more than 1 year to make things much easier for our users. So now pipelines in FCS Express looks like this. Okay, so those are all the building blocks. And the pipeline is executed step by step. And this combination of steps is done by you. So this is what we are going to do in the next few minutes. Okay, so if you have any question, just let me know. So what we will do will be to first create the pipeline, meaning this is what we called main pipeline body. And then we're going to add steps. Okay, maybe we will add downsampling, and then a scaling, and then a UMAP. Okay, so something simple, okay, but very useful and user-friendly. Now, you can select, you can play, you can grab any of the steps available in FCS Express. We have more than 60. Okay, they are well organized in different categories, like predefined algorithms, downsampling step, dimensionality reduction step, clustering step. Okay, you can combine them as you want. So let me close this and let me open, okay, this layout. This is our old friends. Remember, it's a layout in which I already have my cleanup gates and cleanup plots. Okay, and as you can see here at the bottom, I already created a new page. I call it page 1. In this page, I just have one single plot. It's already gated on the cleanup DNA 2, which is the last gate of my cleanup strategy. So this is fully cleaned. Okay, so first thing to do, let's create a gate to identify the population of interest, the population on which we want to run the high dimensional data analysis. Let's say that we want to focus on CD3. Okay, it's just an example. Of course, you can focus on whatever you want. You can focus on T cells, B cells, all cells, total blood, whatever. Okay, so let me call it CD3. Let's do it red. And now I will open a plot and I will keep it there. It's just a plot displaying CD3, nothing special. But this is the plot on which I will display my high dimensional data analysis results. And then we move to tools, transformations, and we are going to create the pipeline. You can click on this little plus and you see we have this option, pipeline. Do not care about the other option at this stage. You see we have k-means, we have SPADE, t-SNE. Those are the old ways of computing these algorithms. Those are the standalone algorithms. So if you run SPADE, in this case, either you run SPADE or t-SNE or PCA or k-mean. This is the old way of doing this type of analysis. We are still keeping them just for compatibility with all the layout. Okay, but nowadays people just go straight to pipeline. And this is the main pipeline body. You can change the name. You can set here the gate of interest. You see I'm selecting my CD3 gate. And here you have all the parameters selected. So I want to use as a initial input for my pipeline every parameters. So my suggestion is do that, because at every step later on, you will be able to select a subset of parameter. So in this main pipeline body, just select all. And indeed, we are automatically selecting all. But otherwise, you can also do right click and select all manually. Okay, so from this plus menu, you see I can access the all the steps. You see we have predefined algorithms. Those are mini pipeline, I would say. We have FlowAI, FlowCut, FlowSum, SPADE. Then we have single step, downsampling. You see, you can add a gate downsampling if you want to narrow down. So now we are starting with the CD3. Maybe at some point in the pipeline, at step number 27, you want to narrow down your calculation to a subset of CD3, you can do that. Then we have internal sampling. We have random sampling. We have a lot. We have target density downsampling. This is the density downsampling invented by SPADE years ago. Okay, then we have dimensionality reduction, PCA, t-SNE, optimized t-SNE, UMAP, okay, clustering, okay, and a lot. I want to start with a mathematical scaling step, okay? This is how do we want to scale parameter, okay? In this case, I'm going straight. I'm going to, for example, select all the CD marker, let's say, giving for granted that we want to use all of them, but of course it's up to you. This is one of those steps in which I was saying you can select what you want. And let's see if we are missing something like this TCR gamma delta, no, bits no, and whatever, like HLADR, IgD. So I probably selected all the markers included in this panel, okay? What this step is doing, it's scaling. So how do we want to scale? let's say that we want to use the arc scene, the very well-known arc scene transformation. Okay. So we can probably spend like half an hour on scaling, but of course we have no time today. So if you have any question, if you have any doubt, feel free to reach us, because it's, I mean, all these, it's a huge word like the high-dimensional data analysis. This is why I want to do something very, very quick and simple today. Okay, this is the first step. Okay, we are scaling the selected parameter. And I already want to show you that if I apply the pipeline, now this pipeline is just scaling. It's not doing any t-SNE, any UMAP. If I apply my pipeline on my plot, okay, you will see that now I have new data in this list. And the data in this case are the scaled, as you can see here, version of all the selected parameter. So you see, step one in my pipeline, it's generating new data. It's generating new data. And we can use this new data for the second step. Okay, so let me switch off the automatic updates in the pipeline. So let's do a pause, and I will add now a downsampling. Let's say interval downsampling. And I will set this to be like 20,000. I just want to do something quick. As you can see, the plots get blank. Okay, this is what we want, because I'm pausing the live updates of the pipeline. So as soon as I do any change, in this case, the change was introducing a new step. Okay, the plot get blank to avoid you seeing non-updated data. So this is done intentionally. Okay, also this arrow, it means there is no statistic to calculate because this plot now it's empty. Okay, and then I can add, for example, t-SNE. Okay, so which are the parameters we want to run t-SNE on? Well, the scaled parameter. So you see here, I should go here and say, okay, I want these. Instead of selecting all of them, if you want to select all the scaled, you can do a filter, okay, and then do a select all, and only the scaled parameter will be selected. And you see here, it's worth mentioning that you have all the control you want. Okay, you can run the exact t-SNE, but probably not a good idea. It's probably better the bar-sat approximation, which is much faster. You can change the amount of approximation. You can change the perplexity. You can change the number of iteration. You can run an optimized t-SNE if you want. Okay, and if you don't want to run a specific step, you can uncheck it. Okay, now t-SNE is there. I didn't delete it, but it's not running. Let me also add another one like UMAP. Okay, let's do the same. I will select all the scale parameter. You see here, you also have a bunch of options like the number of neighbors and the minimum low distance. Again, UMAP as t-SNE, it's an iterative process, so you can set a maximum number of iteration here. Sorry, not a maximum number, it's the number of iteration and so on. And remember, every time we are running a step, we are introducing new data. So it's worth mentioning that we also have a parameter removal step. Okay, what does parameter removal do is hiding parameter at the end of the pipeline, usually, that you don't want to see. So for example, I can say, okay, at the end of the pipeline, I will no longer need all the scale parameter. So why should I have here a huge list when I do not really need those parameters? So I can hide them. So I will just remove from the list, okay, all the scale data. And here we go. Okay, so again, just remember you have here a bunch of steps that you can combine together, a lot. So let me just go back and reactivate the live update. So the pipeline will be now calculated, and it will go, let's say, from the top to the bottom. Okay, so the data, the input data will be scaled, then it will be downsampled. You see it's already done. Then it will be UMAPed. t-SNE will not be run because we didn't check it, and then parameter removal. So now in this plot, okay, I can access UMAP 1 and 2 at the bottom. I do no longer see the scale data because I use the parameter removal. Okay, so this is my UMAP. Okay, so this is UMAP. If you want, you can also, let me go back here. I also want to, I think I have like 2 minutes left. I want to show you also FlowSum, which is really, it's really cool. So I will bring FlowSum right before the parameter removal. And I will run FlowSum on the same scaled step, scaled parameter than UMAP. So we will see, we will compare apple and peers. Okay, I don't need the second parameter removal, and let's go back here. So I will access UMAP, but I will also access FlowSum. You can access like UMAP, FlowSum, Disney, Spade, everything. Okay, insert. Let's create a UMAP plot here. Sorry, FlowSum. Here we go. And this is live updating. You see, it takes like, I don't know, 5 seconds for 26 seconds for 20,000 events. Okay, you will probably have like 1 million. It doesn't matter. But let me tell you that FCS Express is probably the fastest on the market. Now, in the last minutes, I want to show you how you can explore this type of density plots of UMAP to define which is which. Okay, so you see in this UMAP plot, I can display, for example, a legend. Okay, let me do this. I want to place it at the bottom. And now the legend says that the coloring is based on count. What about if I change this and I say, okay, I want to see the CD4, okay, coloring. Now I can clearly see that the CD4 cells are located in this region. Okay, what about the CD19? Okay, okay, they're here, if you can see. It's a very, very red here. Now you can create this automatically for multiple color with the multi-plot capability. So I can say, okay, I want to see the UMAP density for multiple markers like CD4, CD8, C11C, 16, 45 array and a row. Or you can select all, of course. And now you have 6 plots, sorry. Okay, displaying all the density distribution. Okay, so I can, for example, from the second plot, I can clearly see that CD8 are here, here, and here. Those are all different CD8. And CD45 array are here, 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 here. Okay, and so on, and so on, and so on. So this is one way of doing this type of stuff. Another way, and then I will conclude, is to use backgating. I didn't show you backgating before. Let me increase a little bit. So if this is a color.plot, let me do it. Okay, now since the CD3 gate is red, all the events on this plot are red. Okay, but you see, if I create a gate, let's say on these islands, and I make this gate blue or green, okay, let's say unknown population. Okay, now I can see that these populations are green. Make sense? So if I go to multiplot and I say, okay, I want to create a bunch of multiplot, and let's select the same CD4, CD8, CD18, blah, 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 blah. I will be able to backgate automatically and immediately on multiple plots, multiple combination of markers. And I can now navigate on my UMAP and change the position of my unknown gate and see where these cells are located. So I can give names. Now, let me just conclude saying that we have much, much, much more stuff. We are running out of time, so we cannot see everything, but we have many other options. We have heatmap, for example, that are really useful. We have graphing tool. We have high resolution picture. We have many, many other algorithms. So FlowSum and UMAP are just few of them, okay? Also in UMAP, also in FlowSum, you see, I can color each let me do it like CD8. You see, I can color each node of the flow sum tree based on the expression of a given marker. So now I know that at the bottom left, I have some red nodes for CD8. Okay, so let me go back here. I just want to conclude saying that our goal at the Novo software is to save your time. We want to provide you with a user-friendly interface that integrates a lot of tools, and we are really keen in developing any request that that comes from your feedback. And also remember, the PowerPoint-like interface, it's really useful because it speeds up the learning curve quite a lot. So you don't need to learn everything from scratch, even if it's a new software. All the basics are there. So if you receive your instrument, you will also receive the license in an envelope. So with that envelope, you can claim your license. You can make it your own license. Feel free to send us an email, or feel free to send your application specialist from Fluidigm an email if you have any question about that. So many thanks. I'm super happy to answer any question you might have.
Thank you so much, Andrea.
Okay, I can see.
Yes,
so
we have
yeah,
some questions. So shall I read them? Let me read them to you. Is that okay?
Yeah, if you want, I can see them on my second screen. So if it's okay for you, I can go through them very quickly.
Okay.
Yeah.
Right. So I will, you can pick on the first question about how to change the scale of X.
Absolutely. So
okay.
Great, thanks. So you can change the scale on a plot by going to Format. So the Format tab allows you to change everything on the plots. And one of the options is Axis. Okay, so you see here Axis. I can say the X-axis, click on X-axis. Now it's arcsine 5. I can change these to any other scale you want. Now arcsine is probably the best. Or you can also keep arcsine and simply change the cofactor, like, I don't know, 10 or whatever, or 2. Okay, so just change this. Now you see that the plot it's live updating. So it happens in real time. This is 10, this is 2. So it's really easy to use. Now the scale of the plot, it's not linked to the scale in the pipeline. So you can test like 10 different scalings on the plots and just use another scale in the pipeline. So they are completely independent. Make sense?
Perfect.
Okay.
Yeah. Connected to this same attendee, he did ask a couple more questions. We can cluster them together. Right?
Yeah. So, okay. To run the analysis pipeline, is there any requirement on my hardware? So it's, I will say my answer is, it's quite boring, because the answer is like the more power you have, the better it is. Like we are super let's say we did a great job in embedding this type of calculation inside FCS Express, which is a huge enhancement. We are like between 2 and 10 times faster than any other software. But still, if you're running on an old computer, it will take longer. So my suggestion is go with the best computer you have in your hands. The calculation, it's mainly about first the CPU, and secondly about the RAM. And then if you see that it's taking like, I don't know, for 1 million, it might take like 1.5 hours to run Disney, or 1 hour or 2 hours, depending on the computer. You can run it while you are going to lunch, or overnight if you want. So feel free to do that. And about how to specify which samples I want to include on UMAP or Disney. So this is a great point. So in this case, thanks for asking. In this case, I merged 5 different sample in this case, and I can discriminate between the 5 different sample this way. If you see here, now I'm loading. Okay, let me go back to the automatic scale. Okay, in this case, we have 5 different sample merged together. Here we go. Okay, this is sample 1, sample 2, sample 3, sample 4, sample 5. So if you have multiple sample, just merge them. Okay, feel free to send me an email if you have any doubts, and then run UMAP, TSNE, FlowSum, whatever on the merged data. So you will have a unique tree, a unique map, okay, for multiple sample. Then you will always be able to say, okay, I want to create a gate on file number 1, file 1, okay, and then apply this gate on my UMAP. And you see, I can change my gate and see which events on the UMAP belongs to that file, as you can see here. Did I answer your question, Colin? And usually you want to include all the samples from that data set. And it's important that the sample have been acquired in a consistent way. So the variability in the expression of the marker should be biologically generated, not technically, of course. So can I trace the parameter setting of each analysis after I get the results? So there is no way to save all those information like the pipeline in an FCS file. So the FCS format, it cannot handle that. So simply save your FCS Express layout. Okay, you save the FCS Express layout together with your FCS file. So for example, this is my folder. Where is it, my folder? No, oh, here is it. You see, this is my folder, okay? And I have my layout here together with my file. And every time I will open it, I will see every setting. No, so the question is only FCS format could be used in FCS Express 7. No, we are compatible with a wide variety of formats. And you can also, just to mention, you can also load CSV file if you want. So we are really flexible. So FCS, it's not the only file format that you can load. Can one plot on page 1 be connected with plot on another page? That's already done. Okay, so I can show you. I can open up page number 2. I can view 2 pages at a time. Okay, let's do the following. On the left, I have page 1. And at the right, I have page 2, which is now empty. Let me show you. I will, for example, open a plot by drag and drop. Okay, now I will display here UMAP. Okay, so here I want UMAP 1 and UMAP 2. Okay, this is page 2. You can see if I move the gates on page 1, the plots on page 2 is updating live. Doesn't matter how many page, how many plot, they are all connected together. You don't need to do anything, it's done automatically. Any further question?
I think we have done all the questions. I just want to double check. Did we have a chance to look at how we're going to compare healthy versus disease?
Yeah, so it's basic. So as I mentioned, usually concatenate files, right? So in your merged file, you will have control, treated, like day 0, day 1, day 2, whatever. So I can, for example, display file number 1 on this UMAP. Okay, let's do it on one page so it will be easy to follow. View one page. Okay, so these are my 5 different files, and in this case I'm displaying UMAP for file number 1. I can have a second UMAP plot, and I can create a second gate here, and I call this file number 2. Okay, and now I can apply file number 2 here. So you can do this if you want to create a nice picture. Okay, there are also other ways in which you can automatically create this type of gates. But again, we have so many stuff. You see here, I can display my 5 files as a virtual multi-well plate. In this case, I have 5, so I have a 5-well plate. You see well 1, well 2, well 3, well 4, well 5. Okay, each well, it's a file. Then I can create a gate on a single well. this will be file number 1, or let me create a gates on all. And I can say create individual gates for each well. So this will be my file gate. Okay. And now I have automatically created. This is great if you have like 10, 20, 50, 100 files. You see a gate for file number 1, a gate for file number 2, file number 3, file number 4, file number 5. So they are already created. And then you can retrieve statistics for all of them. So there are really infinite ways in which you can proceed with your analysis from this point.
Perfect. Understood. Thank you so much.
Welcome.
Yes. All right. I know it was a tall order to cover a lot of the general steps in getting the data analysis going with high dimensional markers, right? So I know you had to do it really, really quickly, right? And I've tackled on a lot of all the live questions actually already. Teddy, I think you've also helped to handle some of the system-based questions as well, right?
Yeah, just let me answer a quick question from Key. Remember that we wanted to hear some question, just a very short one, if I may. So Key, if you could see my screen, right? So you can see you were asking how do you know they are high quality data. So when it's high quality data, all you need to do is you just see that there's few aggregates, both deep cells and cell. It's very low, very few aggregates. It's a low amount of deep and dead cell. And finally, they have good signal separation in positive and negative population. So you can see that right here with a good signal, and you should know that by here your data quality should go. All right, I hope I answered your question in regards to this area.
A quick look so that we can see, you know, a quick look QC check, right? Before we start putting it into data analysis.
Yeah.
Perfect. Right. So thank you so much. I think we've had our audience spend a nice time this afternoon, really early morning for Andrea.
No worry.
In Europe. But with this, I think we will wrap up today's session. We would probably work closely together with Andrea if we want to take on our next level of data analysis based on the feedback of the participants today to find out if there are additional areas that we can support our high dimensional cytometry users over in our part of the world. All right. So without further ado, thank you very much, everybody, for spending the nice afternoon, a really sunny afternoon for us all together. We look forward to you writing, you know, if you have additional questions. We have at the end of the event a survey. You're welcome to drop in your comments and questions that you might feel that, you know, you sleep on it and you have additional questions. And we will have your local FASs come and approach you and give you the support that you need to be successful in your work. All right. So with that, thank you so much, Andrea, Teddy, and Boon Eng. We will see you guys again real soon.
Bye-bye. Thanks to you, all the
attendees.
attendees.
Bye.
Bye.